We start the first session, Workshop 4, Basic Science and Pathology 1. This session will be chaired by Dr. Jinmo Yang, Dr. Shinji Ito, Dr. Naoshi Nishida, Dr. Akinobu Taketomi. Doctors, please.
Good morning, everyone. So we will now have the first session, the workshop for basic science and pathology. There seems to be a little bit limited number of the participants, so I want to ask everybody to have a deep discussion. So let's move the first talk. The first talk, the title is Immunoglobulin-like transcription factor 2 as an impaired anti-tumor cytotoxicity marker for natural killer cell in patients with hepatocarcinoma. This paper is presented by Dr. Yoshio from National Council for Global Health and Medicine. So could you please start?
Thank you for great opportunity to talk about our work. We have no COI. NK cells function is regulated by various activating and inhibitory receptors. NK cells play an important role to anti-tumor immune responses. NK cells in the peripheral cell, 10% exist, but in the liver, 30% of immune cells are NK cells. Cytotoxicity is reduced in patients with HCC compared with healthy volunteers. And NK cells in cancerous region exhibit decreased frequency, cytotoxicity, and interferon gamma production compared with those in non-cancerous region. However, the impact of HCC on antibody-dependent cellular cytotoxicity, ADCC, is unknown. ADCC NK cells express Fc gamma receptor. Antibodies can bind to antigen on cancer cell, and Fc gamma receptor on NK cells can bind to antibodies. And after binding, NK cells activate and produce perforin and glycozyme and lysis the cancer cell. This is the ADCC. Today's study is aiming to explore potential targets for immune intervention by revealing the surface marker associated with dysfunction NK cells in patients with HCC. We examined peripheral blood mononuclear cells in healthy volunteers in age with from 21 to 82 to examine the influence by aging to immune cells. And after that, we compared HCC patients and age-matched healthy volunteers, and we also examined intrahepatic lymphocytes. We used mass cytometry and examined 39 parameters. This is the gating strategy of NK cells. NK cells were divided into two subsets. NK-DIM subset is today's topic. NK-DIM subsets have high cytotoxicity and express CD16. CD16 is Fc gamma receptor. With aging, frequencies of NK cells were comparable in all NK cells, but about NK-DIM cytotoxicity cells increase with aging. And the changes in surface markers on NK-DIM cells with aging, you can see maturation marker increase with aging, and the activation marker CREX7 160 NKP46 decrease, while inhibitory receptors including PD1, LAG3, ILT2 increase with aging. So to examine the impact of HCC on immune cells, we must compare HCC patients and healthy volunteers with the same age. So we checked the impact of HCC with the same age patient and healthy volunteers. In peripheral cells, NK dim cells decreased in HCC patients, and in liver, NK dim cells were decreased in the cancer tissues. And we found ILT2 upregulation in peripheral HCC patient and also in cancer region. So we focused on ILT2 in this study. ILT2, the receptor of ILT2 is HLA-G, and we first checked HLA-G expression on primary hepatocyte. On primary hepatocyte in cancer region, HLA-G expression were upregulated compared with non-cancerous region. So in cancer tissue, ILT2 expression increase in NK cells, and the receptor HLA-G on tumors are observed. And we checked phenotypes of ILT2 positive NK dim cells. ILT2 NK dim cells expressed inhibitory receptors and decreased NKp46 and CD160 activating markers. And then we checked what induced ILT2 on NK cells in cancer lesions. We co-cultured HCC cell line HUH7 with and without cell contact. Regardless of cell contact, HCC cell line induced ILT2 on NK cells. And the other cell lines, PLC, PRF5, and HLE also induced ILT2 on NK cells. So then we checked few more factors in the supernatants of co-culture. In the few supernatants of HUH7, MIF, macrophage inhibitory factor, is the most abundant factor. And also in the other two cell lines, MIF was the most abundant humoral factor. So we checked MIF can induce ILT2 on NK cells. Recombinant MIF induced ILT2 on NK cells in a dose-dependent manner. And the ILT2 MIF's receptor is CXCR4, and we checked CXCR4 on NK cells. CXCR4 was positive on NK cells. So neutralization of MIF and CXCR4 both inhibited ILT2 induction. So HCC cells induce ILT2 on NK cells via MIF CXCR4 axis. And the HCC patient, NK cells were dysfunctioned in cytotoxicity and ADCC. And we checked ILT2 positive NK DIMS function. ILT2 positive NK cells were all NKp46 negative. So we divided NK DIM cells into three subsets: ILT2 positive cells and NKp46 positive and ILT2 negative cells, and NKp46 negative and ILT2 double negative cells. First, cytotoxicity. Cytotoxicity was decided by the expression of NKp46. NKp46 negative subsets were decreased in cytotoxicity. And moreover, about ADCC, not NKp46, but ILT2 expression indeed decrease ADCC. So ILT2 is associated with the decrease of ADCC in HCC patients. Functional recovery of impaired ILT2 NK dim cells were checked. We neutralized ILT2 by antibody. Cytotoxicity and ADCC both recovered. So ILT2 is the functional marker for dysfunctional NK cells. This is the summary. In HCC patients, on NK cells, ILT2 expression increased, and the cytotoxicity and ADCC decreased. Neutralization of ILT2 antibody recovered cytotoxicity and ADCC. So I want to conclude that ILT2 may be the immunotherapeutic target for HCC in future. We thank for Toshihiro Sakata by Hokkaido University and Yoshihiro Ono in Cancer Institute Hospital. Thank you.
So the professor usually talked about the promising result regarding the role of ILT2 for the treatment of HCC. So this paper is now open for question. Is there any comment? Please.
Dr. Yoshio, thank you for your beautiful data. I'm Jun Arai from Aichi Medical University. So I have two questions. First one is about ILT2 positive NK dim cells. These subgroups decrease the ADCC. What is the precise mechanism of that? I'm thinking about the CD16 level will be decreased, and it's a kind of the mechanism why this IL2 positive NK dim cells will decrease ADCC.
Thank you for your question. These ILT2 positive subsets are all NKp46 negative, but compared with ILT2 negative NK cells, ILT2 positive NK cells more functionally dysfunctioned. So we checked another characteristics of these ILT2 positive NK cells. Body P plus, because oxidation, excessive oxidation is happening in ILT2 positive NK cells. We are now working on this point. Thank you.
Yes, in terms of ADCC, CD16, as you pointed out, the Fc gamma receptor is important. So did you also check that, or are you going to check later?
We already checked CD116 expression. And CD16 expression is not dysregulated in ILT2 positive NK cells.
Thank you very much. And another question is about NK dim. You clearly showed NK dim number is decreased in cancer. So what is the difference according to the histology compared to the poorly the differentiated and highly differentiated? Did you check that, or?
Thank you. Your question is regardless of etiology.
Yes.
Regardless of etiology, NK dim cells is decreased in HCC patient.
Thank you very much. Thank you. And the question from the floor or chairs. That's okay. Could you shortly tell about the background of the HCC patient? Is an advanced case or early case? You examine the HCC patient. Is that an advanced case or early HCC?
This is all the cases which were rejected by operation and not advanced.
Okay.
Yeah.
That's okay. Oh, so thank you very much, Professor Yoshio.
Thank you.
So I want to move to the next paper. The title is Novel Monoclonal Antibody and ADAM17 enzymic inhibitor could induce NK cell-mediated cytotoxicity in ACC by targeting NKG2D ligand. The paper is presented by Professor Jin Arai from the Aichi Medical University. Could you start, please?
Yeah. Good morning, everyone. It's a great honor for me to talk about this topic. So, generally speaking, a recent advancement in the field of HCC chemotherapy for HCC has achieved the many development and merit to HCC patients. However, at most, the response rate is 30%, and more than half of the patients cannot get the merit with this development. So the cancer cells are known to escape from the immunological system by T lymphocytes by partially downregulating expression of MHC class I molecules. Alternatively, NK cells can respond to cells which lack the expression of this molecule, resulting in lysis to the cancer cells, which are resistant to T lymphocytes. Interestingly, a recent meta-analysis showed the percentage of infiltrated NK cells in the HCC tissues was correlated with patients' better outcome. A lot of evidence has been accumulated about the NKG2D, NK stimulating receptor, and its corresponding ligands. In a previous study by Professor Naoya Kato showed the RS2596542 has been detected as a susceptibility gene to HCC among the chronic viral hepatitis patients. This gene, detected by GBAS, is located in the promoter region of MICA. MICA stands for MHC class I related chain A. The function is briefly shown in this figure. As shown here, no expression is confirmed on the surface of normal hepatocyte. However, stress including the viral infection or cell transformation, this type of kill me signal will be induced on the surface of hepatocytes. It can be detected by NK activating receptor and eliminated by NK cells, resulting in less possibility to cause HCC. On the opposite, AA allele patients cannot express more MICA, so easily probable to cause HCC and its spread. The detailed, please take a look at in the detail in this portion between the NK receptor and corresponding ligands. Among various receptors, the principal activating receptor is NKG2D and its ligands, MICA, BICB, and so forth. So for the better outcome of the patients, more expression of these ligands are important. However, it is known that this kind of NKG2D ligand on the membrane-bound form is known to be shed in the tumor microenvironment, including some ADAM protein, to produce soluble form, which function as a decoy in the serum of the patients. So the important strategy against HCC is to stop its shedding to achieve both of them. So for example, when we administer ADAM inhibitor, this shedding will be stopped and more membrane-bound NKG2D ligand will be produced, easily attacked by NK cells to eliminate HCC. Other group from Mount Sinai, Dr. Ferrari Lucas, found, according to the structure of MICA and MICB, alpha-3 domain is known to be the shedding portion. So he newly developed 7C6 monoclonal antibody, which attached to this portion to stop its shedding. As shown here in the data, according to melanoma cells, the 7C6 antibody dose-dependently decreased the shed MICA and increased surface membrane-bound MICA and MICB. Importantly, they also checked in the in vitro NK cell co-culture system 7C6 antibody significantly induced specific killing. Therefore, this monoclonal antibody inhibit this shedding in melanoma cells and increasing cytotoxicity. He is my previous principal investigator when I visited Mount Sinai, so we are now currently collaborating with him. So this study is evaluate the inhibition of MICA and MICB shedding in human HCC by this antibody and ADAM17 inhibitor. We also newly developed in vitro ADAM70 assay system to find and screen some medicine among FDA approved drug library. So firstly, we knocked down each protein which are known to be active in human ADAM and similar MMA proteins. Among them, ADAM17 as well 9 and 10 has a function to shed MIC-A. And in terms of MIC-B, ADAM70 contributes to its shedding. So after knocked down with siADAM17, this protein will be induced, shown in red. So particularly, knockdown of ADAM70 suppressed this molecule shedding. So ADAM17 could be most probable candidate to stop its shedding. We screened with the in vitro assay system. Among 636 FDA drug library, romofungin, a kind of the antifungal drug against yeast, was discovered to suppress its enzymatic function. Further analysis showed its dose-dependent suppressive effect of ADAM17. So administered to the human HCC cell lines, it decreased its cleavage and also increased membrane bound type of this molecule shown in yellow, which was abrogated if pretreated with siADAM17. So this data show romofungin suppressed MIC-A and MIC-B shedding by ADAM17. Because of the time limitation, we could not talk about all of them, but not only ADAM17, but also ADAM9 and 10, we have already screened many medicines and find out some candidates to be probable for the next promising medicine in terms of the ADAM suppression. So this study is currently undergoing, but 76 protein also suppress its shedding, not only MIC-A, but also MIC-B in several cell types to decrease the soluble MIC-B level, whereas when treated with 30 microgram per ml, it also induced MIC-A and MIC-B protein level on the membrane bound type. So next conference, I'd like to talk about the upcoming NK co-culture system data and also the immunodeficient in vivo mice model experiments. So in conclusion, romofungin could work for the immunological control to target the modulation of NKG2D ligands by a suppression of the ADAM17. And 76 also inhibit this molecule shedding to potentially induce stronger NK cell-mediated cytotoxicity. Thank you for your attention.
Thank you, Professor. He did the detailed analysis and tried to inhibit the ADAM17 to recover the NK functions. This paper is now open for question. Is there any comments or question? So I have one question. You show the two options for the enzyme inhibition with ADAM17. One is an antifungal drug and another is antibody. So in terms of the clinical setting, which do you like better, the drug or the antibody for recovering the NK cells?
Yeah, thank you for your question. So there are two options as pointed out, the small molecules to stop the enzymatic function, and another is a monoclonal antibody. The monoclonal antibody is just studied to analyze, so we don't know the schedule, how many amount and the interval, so it's not currently available. But another option to small inhibiting molecule is possible because we could detect among the FDA drug library. So it's more, we can take the shortcut with that.
Yes, okay, thank you very much. Probably the drug is currently under investigating, so it might be better for use, but actually the antibody is less side effect. So it might be better. Is there any question? Dr. Yoshi, please.
Thank you, your presentation. I want to know the expression of ADAM70 is different between intrahepatic and peripheral on NK cells.
Thank you for your question. So we did not check the effect, the expression on NK cells itself. So we needed to check later.
Yeah.
Yeah.
Thank you.
Thank you.
Thank you, Professor Ai, for your kind talk.
Thank you very much.
Next speaker is Dr. Suzuki. Please start.
Thank you. Today, I'm talking about our research, fibroblast growth factor inhibition by molecular target agents mitigates immune-suppressive tissue microenvironment in hepatocellular carcinoma. We thank to President Professor Ueno and chairs for this session for giving me this opportunity to present. There are our disclosure of COIs. Combination immunotherapy, immune checkpoint inhibitors plus molecular targeted agents have been approved for treatment of advanced HCC. However, owing to its relatively low anti-tumor effect, sequential therapy following ICI's treatment is required. The tumor immune environment time, a state of immune competent cell infiltrating the tumor microenvironment, correlate with the therapeutic efficacy of ICIs. Therefore, regarding ICI therapy, the transformation of immune cold tumors, in which immune cells hardly infiltrate, into immune hot tumors, in which immune cells easily infiltrate, is believed to enhance the anti-tumor effect of ICIs. So, to generate a basic rationale for suitable MTAs for combination with ICI, we aim to evaluate the effect of MTA on time using an immune syngeneic orthopic HCC mouse model. First of all, we established immune syngeneic orthopic HCC mouse models in occluded Hep55-1.C cells into the left bilateral lobe of BRAC6 mice and treated them with each MTAs for 2 weeks. This HCC cell line is derived from BRAC6 mice. We chose 2 MTAs as HCC treatment first-line therapy, sorafenib and lenvatinib, and cabozantinib, regorafenib, and ramucirumab we chose as second-line HCC treatment. And also, we chose AZD4547 as FGFR1234 inhibitor. To assess the time alternation, we evaluate the time by immunohistochemistry as follows: tumor surface antibodies. First, we assess the anti-tumor effect of these MTAs. The tumor volume and the number of tumor blood cells shown as CD31 positive cells were significantly decreased in MTAs. And this slide shows the alternation of time by lenvatinib and sorafenib treatment. The number of CD3, CD8, and F480 and FOXP3 and CD11C in sorafenib group decreased, whereas CD8 and the granzyme B positive cell in lenvatinib treatment group increased. Next slide shows the alternation of time caused by ramucirumab, cabozantinib, and regorafenib treatment. F480 and FOXP3 positive cells commonly decrease in these MTAs, whereas in the cabozantinib group, as shown in the red square, granzyme B positive and CD11C positive and NK1.1 positive cells increased. So cabozantinib might activate the innate immunity system. We next assess the immune checkpoint molecules, including PD-1 and PD-L1. So all MTAs decreased the expression of PD-1, whereas lenvatinib and VEGFR2 ramucirumab treatment significantly increased the PD-L1 expression in time. Here, we show a short summary. In all MTA, the number of FOXP3, F480, and PD1 positive cells decreased, whereas CD8 positive T cells increased in lenvatinib treated group. Additionally, granzyme B positive cells were increased in lenvatinib, ramucirumab, and cabozantinib group. So we saw the lenvatinib treatment altered the time from cold to hot state. Each MTAs have relatively specific inhibitory activity. For example, sorafenib and regorafenib has RAF and PDGF signaling, and ramucirumab inhibits VEGF, cabozantinib inhibits MET signaling, and lenvatinib inhibits relatively specific FGF signaling. So we had consequently performed in vivo experiment using FGFR1234 inhibitor. So in the FGFR inhibitor group, the number of FOXP3, F480, and CD11C positive cells decreased. On the other hand, CD8 and granzyme B positive and PD-L1 positive increased. So similar to lenvatinib group, the number in CD8, granzyme B, and PD-L1 positive cells increased in the FGF inhibitor group. So to elucidate the underlying mechanism, we performed RNA sequencing for the tumor tissue in the vehicle treatment, lenvatinib, and FGFR inhibitor group. Here we show the result of RNA sequencing. However, lenvatinib or FGFR inhibitor treatment didn't commonly alter specific immune-related genes, such as T-cell receptor, antigen processing and presentation, and so on. So subsequently, we conducted gene ontology analysis using the commonly down or upregulated genes. The number of commonly downregulated genes was 161 genes, and we use this gene for gene ontology enrichment analysis. These enriched geoterms include retinol metabolism pathways, and arachidonic acid metabolism, retinoic acid metabolism. So several enriched genes were related to metabolic pathway signaling. So several studies which evaluated the immunomodality effect of lenvatinib in real-world clinical practice have shown that lenvatinib might alter the time to immune hot by increasing in cytotoxic T cells and decreasing in T cells. Gene ontology analysis showed that the main underlying mechanism of the FGFR signaling blockade is inhibitory effect of metabolic pathway signaling. Higher baseline PPAR gamma expression is known to associate with short survival of ICA-treated patients in multiple cancer types. In addition, the depletion of energy, e.g. lipid, in time is known to promote the formation immune core environment, especially affecting T cell function, such as exhaustion. It was suggested that suppression of lipid metabolic pathways by FGF signal inhibition, which we were in this study, lead the time to an immune hot state through the releasing of energy depletion in time. So in my conclusion, in orthopic mouse model of HCC, lenvatinib and FGFR inhibitors, which commonly inhibit FGFR signaling, alter the time to an immune hot state by downregulating metabolic pathway signaling. Our findings support the therapeutic concept of combination immunotherapy, including MTAs and ICIs. Thank you for your attention.
Thank you for your excellent presentation. Any comment on audience?
So could you tell me the mechanism by which the in downregulation of the lipid metabolism could result in the immune phenotype of the HCC? Could you tell me the mechanism by which the downregulation of lipid metabolism could lead to the immune hurt?
Thank you for your question. We didn't check the mechanism, specific mechanism of FGF and metabolic mechanism, but by the literature, FGF signal and intracellular pathways of interferon gamma signaling is caused the upregulation of interferon gamma cause metabolic metabolic signaling.
In your data, regorafenib increased number of M2 macrophage.
Yes.
What do you think of this data?
Thank you for your question. Because regorafenib relatively strong effect of PDGF signaling inhibitory, so this may change the M1 to M2 polarization.
Thank you. Other question? Thank you, Dr. Suzuki.
Thank you.
Next presenter is Dr. Kong. Please start your presentation.
Thank you. Thank you very much. Thank you for this opportunity to talk our study today. We have no COI. Development of hepatocellular carcinoma is an important prognostic factor for metabolic dysfunction associated with steatohepatitis, MASH, the same meaning as NASH. However, no preventive method for MASH carcinogenesis has yet been established. This slide shows laparoscopic image and other staining pathology of MASH stage 2 patients. And some patients progress to liver cirrhosis and even hepatocellular carcinoma. We have reported that amino acid glycine has a protective effect on various pathological conditions. For example, glycine suppressed activation of Scupa cells, or glycine suppress of apoptosis of liver sinusoidal endothelial cells. Recently, we have demonstrated that glycine containing diet improves steatohepatitis in experiments using the obese diabetic KKRA mice. The glycine ameliorated insulin resistance and improved steatohepatitis, decrease the elevation of ST and LDL levels. Hepatocyte-specific PTEN, the tumor suppressive factors, develops spontaneously into steatohepatitis followed by hepatocellular carcinoma. Then, therefore, I am in this study was to investigate the effect of amino acid glycine on MASH-related HCC using hepatocyte-specific PTEN knockout mice. Male PTEN knockout mice fed a normal diet or diet containing 5% glycine for 2 or 24 weeks. Wild type or TG negative mice fed a normal diet were used as control. In the short time of steatohepatitis phase, as expected, the PTEN knockout mice spontaneously developed steatohepatitis with varying hepatocytes. Glycine-containing diet clearly improved steatohepatitis. And the elevation of serum ST and LT levels also was significantly decreased by glycine-containing diets. Oxidative stress was visualized by 4-HNE immunohistochemical staining. The glycine-containing diet and clearly decreased oxidative stress in PTEN knockout mice. And the expression of inflammatory cytokine TNF-alpha was also decreased significantly and prevented by glycine-containing diet. The antioxidants such as catalase, SOD1, and glutathione levels was decreased in PTEN knockout mice, and glycine-containing diet reversed the expression of antioxidants to almost normal control levels. In the phase of, in later phase, the steatohepatitis-related cancer phase, the glycine-containing diet prevented steatohepatitis and clearly so far. And interestingly, the glycine-containing diet completely prevented the development of liver cancer. As shown in this graph, the glycine-containing diet significantly, almost completely prevented the development of cancers over 2 millimeters. The expression of IL-12B messenger RNA and interferon gamma and messenger RNA also was increased by glycine. And finally, the CD8 positive cells also was increased in the liver of PTEN knockout mice fed glycine-containing diet. The clearly the CD8 positive cells were increased. This is our working hypothesis. The glycine increased antioxidants and for preventing ROS generation, and prevented the inflammation in PTEN knockout mice. And in the in cancer phase, glycine increased, and the expression of IL-12B followed by the increase of CD8 positive cells. This effect prevents carcinogenesis. In conclusion, glycine improves steatohepatitis and inhibits hepatic tumorigenesis in hepatocyte-specific PTEN knockout mice. And the glycine acts as antioxidative stress to improve steatohepatitis in the early phase and contributes to the activation of anti-tumor immunity and suppresses tumor formation in the carcinogenic phase. In conclusion, the glycine has the potential to prevent MASH-related carcinogenesis. Thank you for your attention.
Oh, thank you for your presentation. Dr. Hikita, please.
Thank you for nice presentation. And so my first question is what is the mechanism of the ALT decreasing by the glycine feeding? So you mentioned that so oxidative stress decreasing by the other antioxidant of the glycine. So you think that the hepatocyte apoptosis or cell death are directly inhibited and suppressed by the glycine, or macrophage activity you mentioned that decreased by the glycine supplementation. So do you think the glycine suppressed the macrophage activity leading to the hepatocyte cell death suppression? Which story do you think?
Thank you for your question. And Actually, as shown in background slides, glycine has many effects for the liver pathology. And actually, I consider some mechanism and involves the improvement of steatohepatitis and in pit-knockout mice. At first, as shown and in my slides, glycine increase the antioxidant glutathione. Actually, the glycine itself is the material to produce glutathione. It's the first mechanism.
Okay.
And we already shown that glycine directly prevents the activation of Kupffer cells. And both mechanisms, the direct and antioxidant effect for hepatocyte and the prevention of the inflammatory cells and ameliorates the steatohepatitis.
Thank you. So you mentioned that two possible pathways
Yes.
associating and mechanism. Okay. And the next question is that you've shown the steatosis is also decreased by the glycine. So what is the mechanism of the glycine, by the glycine decreased steatosis?
And very nice point. Then actually, I don't have so good data to show to answer the question. We already shown that glycine and improves insulin resistance and using KKY mice model. But in pit-knockout mice, there is no insulin resistance. And I can't answer the same mechanism in this model. Then I measure some lipogenesis or lipolysis related gene, but so far I don't have so much good data to ask to answer the question. Sorry.
Okay. Anyway, very interesting. Thank you so much.
Please.
Thank you for your excellent presentation. I am Coach Miura from Jichi Medical University. So we are now investigating NASH using a pit knockout mice, as you know. So my question is, so in my data, most of treatment agents that have positive data show suppression or improvement in background NASH. Do you have any data glycine directly suppress the tumor proliferation or growth?
Thank you for your question. Unfortunately, I don't have the direct. I don't have data directly to answer the previous question, but it has already shown that the glycine isn't cell toxicity. It hasn't cell toxicity. Even the glycine is the metabolite is essential for the tumor cell progression. Therefore, I don't think the glycine directly inhibits the cell proliferation in tumor cells. In this study, I show the glycine activates the anti-tumor immune response. Then I consider the glycine prevents the tumorigenesis by the activation of the anti-tumor immune system.
Thank you.
Dr. Arai, short comment, please.
Thank you so much for beautiful data. So it's fascinating. Glycine prevents carcinogenesis as well as steatohepatitis. So you mentioned about the inflammation-related molecules like IL-12, interferon gamma between the glycine with or without glycine in PTEN knockout mice. So I'm asking short question about the tumor-related genes between with or without glycine in PTEN knockout mice. Have you checked between these two tumors about the tumor-related genes?
Tumor-related genes.
With or without glycine, yes.
And sorry, I haven't checked it. Then I need to do it later.
Thank you.
Thank you, Dr. Ko.
Thank you very much.
Now we have last speakers from internal medicine of University of Yamanashi, Professor Yu-Hong Ma. Professor Yu-Hong Ma. Professor Ma is there? There is maybe the cancels of his lecture. So now we have closing of this session. Thank you for thank you for all the speakers and the participants. Thank you very much.